In a development once deemed beyond the reach of science, a team of researchers has accomplished what many thought impossible. They have built the first ultra-precise, three-dimensional map of a mammal's brain at an astonishingly detailed level. Using a cubic millimeter of brain tissue from a mouse, roughly the size of a grain of sand, scientists have captured the intricate architecture, neural activity, and connectivity of 84,000 neurons and over 500 million synapses. What emerges from this monumental effort is not just a technological triumph, but a new foundation for understanding the brain with a clarity and comprehensiveness previously unimaginable. The achievement is the result of nearly a decade of collaborative research involving 150 scientists from 22 institutions, spearheaded by the Allen Institute for Brain Science, Baylor College of Medicine, and Princeton University. What they've created is more than just a map. This is a living, breathing window into the brain's inner universe. It's as though researchers have opened a new galaxy for exploration, one made not of stars and planets, but of pulsing neurons, swirling connections, and the electrical dance of cognition. To appreciate the scale of this accomplishment, consider the data. Just this one speck of brain matter yielded 1.6 petabytes of information, the equivalent of 22 years of continuous high-definition video. This data isn't locked away behind institutional walls. In a move that will undoubtedly accelerate further discoveries, the entire dataset is publicly available, opening the door for researchers across the world to explore the brain's secrets with unprecedented ease and speed. The project, part of the Machine Intelligence from Cortical Networks, Microns, initiative, involved more than just imaging. It started with recording brain activity in a live mouse using advanced microscopes while the animal was awake and actively processing visual information. Scientists played scenes from movies like The Matrix and Mad Max, Fury Road, as well as adrenaline-charged extreme sports clips. This real-time stimulation was crucial to capturing how neurons fire during sensory experiences. Following this, the same piece of brain tissue was painstakingly sliced into 28,000 ultra-thin layers, each thinner than a human hair, and then reconstructed into a high-resolution 3D model. This stage alone required 12 days of continuous machine operation with human supervision to avoid the loss of any critical sections. Once digitized, teams at Princeton deployed machine learning tools to trace and segment individual neurons. These AI-generated maps were then reviewed and refined by human scientists, ensuring scientific accuracy without losing the beauty of computational efficiency. What resulted is a full connectome of the mouse visual cortex, a comprehensive map of how neurons are physically and functionally connected. And while this only represents 1/500 of the entire mouse brain, the detail is staggering. Each neuron, each axon, and each synapse has been identified, colored, and contextualized. For researchers, this is the equivalent of being handed a circuit diagram of the most complex biological machine in existence. Perhaps even more exciting is how this new knowledge opens doors to previously unattainable research. For the first time, scientists can examine how specific types of brain cells interact to perform complex functions. This could radically transform how we approach neurological and psychiatric diseases like Alzheimer's, Parkinson's, autism, and schizophrenia, disorders all linked to communication failures in the brain's network. By comparing diseased brain wiring to the now available healthy connectome, researchers may soon be able to identify and fix disruptions with far more precision. This achievement also shifts neuroscience firmly into the digital age. As Dr. Sebastian Sung of Princeton University aptly put it, the connectome represents the beginning of the digital transformation of brain science. With a few keystrokes, researchers can now retrieve insights that once would have taken an entire doctoral dissertation to uncover. The tools of discovery are faster, more accessible, and more interconnected than ever. This moment also redefines what's possible in science. In 1979, Nobel Prize-winning molecular biologist Francis Crick wrote that mapping a cubic millimeter of brain tissue in full detail, down to the firing patterns of neurons, was an impossible request. Today, that so-called impossibility has not only been realized, it's been made open to the world. Crick's imagined limits have been shattered not by isolated breakthroughs, but by sustained collaboration, technological vision, and a profound belief in the value of exploring the unknown. The broader implications of this work extend far beyond mice. Lab mice are already key models for understanding human biology, and the similarities in brain structure offer a strong foundation for translation. As scientists refine the techniques used here, they edge closer to mapping larger portions of the brain and, eventually, aspects of the human brain itself. While mapping the entire human connectome at synaptic resolution remains a challenge of immense scale, researchers now believe it's not a matter of if, but when and how. The task ahead is still formidable. The human brain is more than 1,500 times larger than the mouse brain, posing massive technical and ethical questions. But the groundwork has been laid. Researchers already envision tracing long-distance axons through the human brain, offering a middle ground that preserves rich connectivity data without requiring impossible levels of detail. What we are witnessing is the emergence of a new era in neuroscience, an era where the inner workings of the mind can be rendered not just in theory, but in full color, with dynamic interactivity and analytical clarity. This work doesn't just move the goalposts of scientific knowledge, it redraws the field entirely. It shows us that the brain, once the final frontier of biology, is no longer unknowable. It is complex, yes, but it is a complexity we are learning to navigate one neuron at a time. And with that navigation comes a new kind of hope. Hope that by understanding the brain's map, we can better understand ourselves, our thoughts, our memories, our emotions, and perhaps one day, the very essence of consciousness. The brain's secrets are beginning to yield, not to speculation, but to science. And what was once deemed impossible is now part of our shared reality.